Next speaker is another of our faves in the CPFRC, Julie Christianson. Julie is a preclinical pain researcher, and I think that she has, if not the best, one of the best animal models of nociplastic pain, and hopefully that's what you're talking about today.
That is what I'm talking about today.
Good,
yes.
good.
Yes, so I'm here to bring all of the mice. I'm here to talk about critters. Okay, so thank you very much to Dan and to Afton, especially for the invitation to speak today. A lot of this is going to be a repeat. It's been a wonderful introduction to what I'm going to talk about this morning, the impact of early life stress on pain processes and other effects later in life. So a lot of what we know came from this 2012 Kaiser Adverse Childhood Experiences study, where they polled over 17,000 participants in the mid-90s. And they asked them about the number of abuse, neglect, and incidents of household dysfunction that they experienced. And at least 67% of those polled reported at least one ACE, with 87% having more than one, and 1 in 8 reporting 4 or more. And this is important because as these ACEs accumulate during development, it's more likely to manifest as altered behaviors later in life, as well as physical and mental health. And this has also been studied in a lot of chronic functional pain disorders or the centralized pain disorders, including headache, fibromyalgia, gastroenterological diseases, irritable bowel syndrome, and what I'm particularly interested in, the urologic chronic pelvic pain syndromes. And so the way that we've modeled this in my mice, or in my lab, is that we use neonatal maternal separation in mice. So we order in pregnant moms, and beginning the day after they're born, we will take the whole litter and we put them in a beaker with some home bedding material, and we'll put them in an incubator for 3 hours a day. And so they're held at 37 degrees, 50% humidity. They're perfectly fine. They're all together, but they're separated from their mom 3 hours a day for 21 days. And then we wean them, and then we begin testing them around 8 weeks of age. And so I'm obviously interested in urologic pain. You may be asking yourself, how does one test this in a mouse? And so one of the main ways we do this is using pelvic organ distension. So we measure the electromyographic activity of the abdominal muscles, which will reflexively contract in response to distension of a pelvic organ. And this is called the visceromotor reflex, or VMR. And so this is a schematic of our setup. And hopefully you can appreciate this is an example of colorectal distension. As we increase the pressure of the balloon that's inside the colorectum, we get an increase in the activity of the activity in the abdominal muscles. And if we chart this as a percent of baseline activity over the balloon pressure, we get a nice stimulus response function. And so this is great for organs you can access from the outside, colorectum, vaginal, bladder for females, but males are very difficult to catheterize externally, so we also use mechanical sensitivity. A lot of you have probably seen this in hind paw reflexiveness, where we apply graded von Frey filaments to the perigenital area to get a mechanical threshold that's required to elicit a response. So this early work was done by my first 2 graduate students in the lab, Angela Pierce and Isabella Fuentes, and so this is using that visceromotor response. And so, on the left we see vaginal responses, and on the right is bladder. The black lines are naive, so these mice were also ordered in. They were born in-house, same time as everyone else, but they did not undergo the separation. And the white circles are the mice that underwent that separation. And hopefully you can appreciate that we see a leftward shift in that stimulus response function, suggesting that they were more sensitive to both vaginal and bladder distension. In the males, we did the perigenital. So in this case, I have the arrows pointing like which way pain goes up and which pain, like pointing the directionality, because it gets a little confusing. So in the VMR studies, pain means upward means more pain, and in the perigenital, it means downward means more pain, because it takes a smaller amount of force to elicit a response. So they're more sensitive to a lighter touch. And so in our male mice, we saw that they were indeed exhibiting perigenital hypersensitivity. Interestingly, we did not see this in the colorectum, which was interesting, and it took a lot of convincing for people in the field to believe me that this was true, because in rats, models of maternal separation, this is usually the main outcome, is colorectal sensitivity as well as altered bowel habits. Okay, so we've established that off the bat, the mice have increased urogenital sensitivity. So now we kind of tinker around with that. So we've done a lot of work using water avoidance stress. So this is an adult stress exposure. Essentially, it's just what you see here. I went to Target and I bought plastic bins and I hot glued Ziploc containers on top of those. So we are very handy in the lab. And you can fill these with water and then you put the mouse on the platform for an hour and they just stress out. This is a unique model in that it causes uniquely mast cell degranulation. And mast cells are very involved in a lot of the functional pain disorders that we've studied. So when we do this, and we again repeat our VMR studies, we see an exacerbation of that bladder response. So after 8 days after that the WAS exposure, we see an increase in the activity. And interestingly, in the colorectal distension, we saw an increase only in the naive. And then looking at the male mice, again, we saw an increase in perigenital sensitivity, but in this time we did see an increase in colorectal sensitivity. So this kind of alludes to those sex differences that have been talked about so far this morning. We see a lot of sex differences in this model. Okay, so that was one way to make things worse. One of the big things we're interested in the lab is how does voluntary exercise improve things? A colleague of mine said this probably 12 years ago. She was in a dissertation defense, and it was a student who was studying exercise, and she asked the student, what do you think your control group is? Is it your sedentary mouse or is it your exercised mouse? And she's like, well, obviously it's the sedentary mouse. And she said, what do mice do in the wild? Do they sit in a cage all day? And she's like, oh, no, actually, the exercised mouse would be the control. So that is more what a mouse would normally be doing. They don't actually live in cages, in the woods. So this really made me think differently about our interventions, and that the exercise, giving them a running wheel, is actually more indicative of what they should be doing. And those sedentary, so the sedentary caging is very American to be sitting around. Okay, so now we will give them, they go through the whole paradigm, but now at 4 weeks of age, they are pair housed either in sedentary conditions or with a running wheel. And in these graphs, the blue are going to be naive, the red are the separated or stressed mice. The open circles are exercise and the closed ones are sedentary. And hopefully you can appreciate that our sedentary mice still had a significant increase in their bladder responses, but the exercise mice were normalized. And this is the same for the male mice. For the perigenital sensitivity, it was completely normalized as well. And so when we're thinking about what the mechanism could be for controlling all of this, we've already heard a lot about the hippocampus. You're going to hear more about it right now. So the hippocampus is a major limbic regulator of the HPA axis, the hypothalamic pituitary adrenal axis. So the eventual product of that axis is glucocorticoids. And the hippocampus expresses a ton of glucocorticoid receptors. And in response to that GR activation, it has glutamatergic input onto GABAergic interneurons that will shut down the hypothalamus. So that is a very complicated way of saying if you have a big, beefy, healthy hippocampus, you're going to have less of a stress response. And it's been shown in preclinical and clinical literature that exposure to early life stress has a significant impact on the hippocampus. So we see decreased gray matter volume in the hippocampus, decreased functional connectivity with other regions of the brain, increased methylation of stress-related regulatory genes, which will ultimately mean a decreased expression level. And this has all been correlated with psychological symptoms. And so in our previous work looking at separation and adult stress exposure and exercise, we have seen that our separation paradigm does decrease stress-related receptor expression in the hippocampus. It's further decreased by water avoidance stress, but it's not affected by exercise. What exercise does is increase BDNF, brain-derived neurotrophic factor expression, which increases LTP, neurogenesis, and all the good things within the hippocampus that makes it beefier. And it's kind of we think it's like a Band-Aid that helps increase the hippocampal output without really affecting what was wrong underneath. And so we've looked at this in a couple different ways. This is looking predominantly at male mice here. So these mice were allowed to run from 4 to 8 weeks of age. At 8 weeks, we injected them with BRDU, which is incorporated into actively dividing cells. And then we counted the number of BRDU-labeled neurons within the dentate gyrus of the hippocampus. And what we saw is that there was a significant decrease in the number of BRDU-labeled cells in the sedentary mice, but exercise significantly increased it in both groups. We thought, great, that was early, let's look at it later. And sadly, not much happened. So at 6 months of age, overall, so if you look at that top, the 3 cells per section, if you look back here, we were more around 10. So obviously there's an age effect where you have a reduction in this neurogenesis as the mice are aging. So again, these are 6 months old. But the one thing that we did notice is that the mice, the female mice, particularly the unstressed female mice, had a higher rate of neurogenesis compared to the males. And this is shown in that sex differences panel there in D. And so we looked at this a little bit further, and we were also interested in the laterality of the hippocampus. Unfortunately, we didn't discern that for the counts. So when we looked at BDNF protein levels in the female and the male, in the female, we saw that there was an interesting laterality and effect of exercise, where the right hippocampus was really responsive to exercise, but only in those unstressed mice. And the males were really flatlined. So if we do a sex comparison for left versus right, we saw major sex differences where the females really do have a more robust level of BDNF as well as increased neurogenesis happening. So that was one study. So we had a pilot study a few years ago wanting to do neuroimaging in our mice. This was really interesting, a lot of the work that has come from here, showing that there are differences in functional connectivity in brains of patients with UCPPS, particularly those with early life stress experience. And so we were using our mouse model to see if we can recapitulate some of those findings. And so this was work done by now a medical resident at the University of Iowa, but he was a postbac at the time. And we were doing MRI and MRS spectroscopy in our mice. And so one of the things that Aaron saw was that the left hippocampal gray matter was significantly smaller in our NMS female mice compared to naive, which is reflected in clinical populations. And so I currently have a graduate student, Tara McQuillan, who has taken this and expanded it. So now we have male and female, naive, separated, sedentary exercise, left, right. If you think about the statistical analyses we have to do on this study, you would know why I need a time machine to go back and make different choices in my life. And so the timeline is to do separation, they exercise, and then we did baseline scans at 5 months, and then we did water avoidance stress, and then another repeat scan 24 hours later. And these are just kind of the general spectroscopy. We don't have the MRI results done yet. We're still tracing brains. But generally the spectroscopy, this is kind of a wide shot. This is before and after water avoidance stress. This isn't taking into account the separation. So you can see there's laterality differences. The left hippocampus is far more responsive than the right. And the male hippocampus has far more significant changes than the female. And one interesting hit that keeps coming up over and over again is glutamate. And so glutamate was significantly altered in the male mice. And these are left and right, female, male, baseline, and post water avoidance stress. And so here's my complicated statistics. So in the left female hippocampus, we saw this great interaction of WAS, separation, and exercise. And then the male was particularly with the water avoidance stress. And in that right hippocampus, we see really significant changes in the male mice, which were really exciting. And this is because everywhere we've looked recently in the hippocampus, it points back to glutamate. And so we did a whole other study with the same number of mice, and I know you should question my sanity, looking at reduced representation by sulfite sequencing to measure DNA methylation. And one thing that really popped up over and over again was glutamate signaling. And so we saw changes in methylation, hypomethylation in all of these glutamate-related signaling or genes, and that my research assistant went in and validated with RT-PCR, and she did indeed validate a lot of these genes were differently expressed. And interestingly, for bassoon, specifically in the exercise group, that's hypomethylated. So exercise did cause a flip in that methylation pattern. And then my last talk on glutamate, we've been looking at mitochondrial respiration in the hippocampus. And so this is a slightly different study. This is now, instead of trying to make everybody with exercise, we're making them worse with a high-fat, high-sucrose diet. And so now the open they're worse, they're not better. But this is looking at hippocampal complex 1, which is a glutamate-dependent respiration. And this was significantly higher in our females, just in general, compared to males, but it was reduced in our separated females, indicating that early life stress exposure made them less metabolically resistant to the stress. Okay. So with all of that, hopefully I've convinced you that early life stress in mice does result in this urologic hypersensitivity and dysfunction. I didn't show you the dysfunction part, but you'll just have to believe me. It's exacerbated by acute stress exposure. We do see common comorbidities. We do see widespread hypersensitivity. We've looked at migraine-like behaviors, hind paw, foot paw. We do see mood disorder. We see altered anxiety and depression-like behaviors. And we are also looking at cognitive behaviors, and those seem to be affected as well. These are linked with hippocampal deficits. We see altered metabolite responses to this water avoidance stress, altered DNA methylation patterns, as well as decreased mitochondrial respiration. And we do think that voluntary wheel running does improve hippocampal neurogenesis and the gene expression, and it can normalize most of these pain-related and mood behaviors. So we believe that an early life stress model does provide clinically relevant outcomes for testing pharmacological or lifestyle interventions that will hopefully help treat widespread pain and associated comorbidities. And I'd like to thank the folks that actually did all of this work, and I'm happy to take any questions.